Very much, and thanks very much for the invitation from the Great Wall Conference. Good morning, Professor Gray, Professor Brenning, Professor Cao, and Professor Liu. And I'm Jun Yu from the Department of Cardiology, Daping Hospital, which was supervised by Professor Chunyu Zeng. But unfortunately, he is not available to make it here, and he sent me to give our progress translational study to share with you guys. And our lab has long been working on the basic and translational study about the hypertension and the cardiovascular remodeling. And we are lucky to have the opportunity to publish our work about the NAT10 mediated AC4C modification in VSMCs during post-hypertension. And this work has been published earlier this year in the European Heart Journal, which I think is a very pleasant experience. And today I would like to share another topic. It's about the epigenetic regulation of the translational transmitted gamete or embryonic originated hypertension. So we all know that the hypertension is the leading risk factor for the cardiovascular diseases. Although we have 5 major types of drugs to control blood pressure, we cannot prevent hypertension before it's overcome. So the mechanism underneath remains to be illustrated, both in all around the world and in China. For the post years of study, it has shown that the genetics is a major driver of the development of hypertension. But however, the GWAS studies indicated that the SNPs that are related with hypertension can only explain less than 10% of hypertension. That is to say, the input of the environment takes a lot of functions. Well, speaking of the environment, the adverse factors exposure, including the disease and the bad lifestyle, or the air, such as air pollution, noise, and they can all contribute to the hypertension, through which we call the epigenetics. That is not change the DNA sequence, but they can significantly change the expression of the functional genes. Well, speaking of the epigenetics, there is a very classic example of the Dutch famine during the World War II. And when the children grow up, born from the Dutch famine, when they grow up in their adulthood, they have a significantly high development of the metabolic diseases, including hypertension, obesity, or diabetes. And following to this phenomena, an English scientist named David Barker raised the theory that the development origins of health and disease, the Doha theory. And after that, this theory can be extended to the gamete stage. That is to say, when the adverse factors stimulates the gamete or the embryonic, they can contribute to the development of diseases in the offspring. So this theory opens the new horizons for the chronic disease, and we take the advantage of this theory and to do our studies in the development of hypertension then. Also, the epidemiology studies suggested that including the Framingham Heart Study, the maternal and paternal cardiovascular disease cohort, or the studies in the born in Guangzhou cohort studies, they all show that these gamete or embryonic derived originated diseases has a trend of intergenerational or transgenerational trend. And we also want to know that whether this kind of hypertension has a trend of inter or transgenerational studies. So we use the animal models for the further study. Before that, I have to make a brief introduction of the intergenerational or transgenerational inheritance. And so for the adverse factors stimulates when it takes place before mating to the male or female mother or father before mating. So the exposures are subjected to the germline. And then when the phenotype transmitted to F2 generation, it was considered as the transgenerational. And when it come out in the first generation, it was considered as the intergenerational. But when the stimulations take place in during pregnancy, that means the baby, the first generation baby, are in the embryo that is directly stimulated by the adverse factors. Therefore, when the phenotype showed in F3 generation, it can be considered as the transgenerational, and when it only showed in F2 generation, can be only regarded as the intergenerational. That's the difference. And so what are the major mediators of the trans or intergenerational inheritance? For now, we believe that major types of epigenetic modifications, including DNA methylation, histone modification, or the non-coding RNAs, can all serve as the mediators, which has been proved in different diseases and in different species. And our work started from the maternal infection, which was a frequent complication during pregnancy. And using this model, we show that when we treat the pregnant mice with LPS, and it can induce the hypertension in the first generation, and along with the impaired sodium excretion. So take advantage of this model. We further constructed the transgenerational rights. As we can see here, that when we treated the pregnant mother with LPS, we can get the first generation of male or female rights that are with hypertension. And we then used the hypertensive male rights to mate with normal. We took the hypertensive female rights to mate with normal tension male rights to generate the second generation, and so on. As we can see that the prenatal LPS treatment exposure can impair the salt excretion and induce hypertension. Unsurprisingly, this phenotype can be transmitted from the first to the third generation. That is a spontaneous hypertension. And since we found that the urinary excretion is impaired, we then treated the fourth generation and fifth generation with high salt diet, and found that these two generations can also develop the salt-sensitive hypertension. And this phenotype was disappeared until the sixth generation. So this is the first model show that the prenatal LPS treatment can induce the transgenerational in rats, transgenerational hypertension. And so what is the mechanism that's causing the hypertension in the offsprings? So we performed the RNA sequencing using the kidney tissue from the F3 generation rats, and we found that a lot of genes that regulating the sodium ion transport was changed. We also validated these genes and found that a known gene called REC1 was significantly upregulated in the kidney of the offsprings, and it is consistently upregulated from F1 to F3 generation, suggesting that it could be a potential functional gene that is responsible for the development of hypertension. And the REC1 has been reported by other groups taking major functions during the development of salt-sensitive hypertension, and the REC1 can activate the downstream MR signaling to induce the impairment of sodium excretion. So we validated the expression of REC1 in each generation's kidney tissue of the hypertensive offsprings, and we also use an inhibitor of REC1 to treat different generations of the rats, and which significantly recovered the transgenerational transmitted hypertension. This data indicated that REC1 is necessary for the transgenerational impaired urinary sodium excretion and hypertension. And since the downstream of REC1 is MR, we also use the spironolactone and MRA to treat the F1 and F3 generations of the hypertensive offsprings. and it also significantly recovered the blood pressure and the urinary excretion. So this data suggested that the MRI could be a potential therapeutic strategy to treat this kind of hypertension. And then we wondered, how can this phenotype be transmitted through generations? What is the mediators? As we know that both genetic and epigenetic can be the mediators. So we first using the DNA sequencing to check whether the REC1 gene has a mutation there, but we found no mutation in the DNA sequence of the REC1 gene. We also performed the DNA methylation detection of the REC1, found there's no altered DNA methylation level in the REC1 gene. So we next try to check whether the histone modification, which is another strong kind of epigenetic modification in the kidney. And we found that the H3K2 bimethylation was significantly changed, and it was decreased from the F1 to F3 generation. And it is known that the H3K9 bimethylation is a repressor of the REC1 gene. So it could be the mediator of the transgenerational transmission. And after that, we wondered what is the enzyme that mediates the histone modification. So we screened the enzyme that is known to induce or reduce the histone modification, and we found that the KDM3B, which is a histone demethylase, that is changed in the first generations, in the F1 generation of the ovarian tissue, indicating that the KDM3B can be the trigger, can be the demethylase that is responsible for the H3K9 modification. And what is the very beginning of? So what is the very beginning of these signalings? And we noticed that after the prenatal LPS treatment, there is a significant oxidative stress in the mother. And so can the ROS be the starting trigger? So we then used the tempo and non-anti-oxidative stress drug to treat the mother after the LPS induction in the mother, and we noticed that all the phenotype was recovered from the F1 to the F3 generation, indicating that the anti-oxidative stress treatment can be a potential preventive strategy to the prenatal LPS induced transgenerational hypertension. And so this study has been published in 2022 in Circulation. And after that, we are wondering, can this phenotype be copied in the gamete-mediated hypertension? So we use another model, that is the PM2.5, which is a fine particle from air pollution. And we previously we found that the PM2.5 exposure to the male rights before mating can induce the hypertension in the first generation. We then, using the same strategy to constructed a cohort of male rights to see whether the gamete exposure can induce the transgenerational hypertension. And surprisingly, we also found that the paternal PM2.5 exposure could indeed can induce the transgeneral inheritance of hypertension, which also induce the impairment of the urinary sodium excretion. And we also performed the in vitro fertilization to prove that this phenotype was transmitted through the sperm, but not other factors. And for the mechanism, we also found that the histone modification could be the mediator that mediated the transgenerational inheritance of hypertension. But this time it's a little bit different from in the mother exposures. we found that the PM2.5 stimulation to the male rats not induced directly influence the function of the kidney, but it influenced the function of adrenal gland, which is an important gland that can synthesize the aldosterone. And PM2.5 really increased the level of the aldosterone secretion from the adrenal gland. So this work, we are still working on these projects, and hopefully we can get this work published soon, and we can share with you guys next time. And after that, our team found maybe there are major problems that needs to be solved in the future. First of all, besides PM2.5 or the LPS, is there any other risk factors that can induce the transgenerational hypertension when they are subjected to the mother or father before mating? And then is there a common mechanism behind this kind of gamete or embryo originated hypertension? If we know the common mechanism, we can just to find the key targets. And then what is the epigenetic mediators along with histone modifications? Is there any other like the non-coding RNAs that can mediate the transgenerational inheritance? So after solving these problems, we think we can bring novel prevention and treatment strategies for the treatment of hypertension in the future. And finally, I would like to thank my mentor, Professor Chunyu Zeng, and our collaborators, including Professor Yibing Wang, Xu Qingbo, Huang Yu, and Professor Zhang Bing from different universities in China. And thanks very much for your attention. I'd like to take any questions. And here's the scenery of our beautiful city, Chongqing. I welcome all you guys to have a visit in the future. Thank you very much.